All right, once the DNA is copied, now we're ready for mitosis. We just have to await the signal. When the signal arrives, the cell begins to pack up the DNA, just like you would pack up your belongings if you were moving to a new home. The loose chromatin that we saw here to make the DNA accessible while the cell was doing its work gets coiled tightly around itself, and then the coils turn into coils. You've probably seen this happen if you've twisted a piece of string and it coils on itself and then on itself again. So that's what's happening to the DNA. Eventually, it gets so tightly coiled that we see this sort of classic chromosome structure that you see here, very dense aggregation of DNA. Now, each one of these strands is an identical copy of the DNA, and they're sort of stapled together through what's called at the centromere. So each of these identical copies is called a sister chromatid. This is the same as this, and the centromere is where they are gathered together so that they don't wander off before we're ready to split them into the two daughter cells. Before DNA replication took place, a pair of chromosomes would have looked like this, a single coiled strand of just one copy of the DNA. After it's replicated, we see the two sister chromatids attached to one another so that we don't lose track of them. So, unreplicated chromosomes, replicated chromosomes. All right, now we're ready for mitosis. There are 5 stages that have sort of major characteristics as the cell divides. While it appears that there are clear boundaries between the two, or between any two phases, it's actually a process. So the boundaries between, say, prophase and metaphase aren't always clear. When the cell receives the signal that it's okay to divide, it's in interphase. Right, so in interphase, we see the loose chromatin, DNA, but everything's been duplicated. The DNA, all of the organelles, and there's plenty of cytoplasm. We also start to see these structures called centrioles, and they're going to play an important role in pulling the chromosomes apart as the cell divides. All right, once we actually enter mitosis, the first phase is called prophase. Prophase is about preparation or packing to get ready for this big change. So, and I'm emphasizing the letter, of course, to help you remember what happens in each one. So the DNA goes from being in loose chromatin form to getting packed into these dense chromosome shapes. The nuclear envelope, which was keeping the DNA all in one place, starts to dissolve and disappear. And then the centrioles move to either end of the cell, and we start to form what are called spindle fibers. The spindle fibers attach the centrioles to the membrane, sort of anchor each centriole to the end of the membrane, and the spindle fibers attach to each chromosome, each replicated chromosome. Metaphase is where the chromosomes are arranged in the middle of the cell, which is referred to as the equatorial plate. So here, the spindle fibers are actually moving the chromosomes, so everybody's lined up in the middle, and each chromosome has 2 spindle fibers attached to it, one that's on this side and one that's on this side. So metaphase is in the middle. In anaphase, the chromatids are pulled apart. The spindle fibers start to shorten, which separates the 2 sister chromatids, and each one is held at the centromere and is being pulled towards the opposite end of the cell. Because chromatids are just, floppy pieces of DNA, they make this characteristic arrow shape that's pointing towards the end. It would be the same thing if you took a piece of thread and pulled it. The 2 loose ends would be dragging behind. So half of the sister chromatids go to one end, half go to the other. You can see the cell is more elongated, oval-shaped here. That's to give the cell room to pull them apart and get complete separation. In telophase, we've now formed two nuclei. So here you can see that the nuclear envelope is beginning to reform around each new set of chromosomes. So we now have two nuclei. And while it actually began in anaphase, we can see that the cytoplasm itself is starting to divide. And that brings us to the phase known as cytokinesis. Cytokinesis is when the division of the cytoplasm completes and we have two fully separate daughter cells. Kinesis means movement. I don't really know how this one was named, but basically each daughter cell is moving into its own space, and that's cytokinesis. All right. Now that we've got our two daughter cells, they enter the first gap phase and start living their lives, and the whole thing starts over again. Want to take a look at some actual microscope images to give you an idea of what you'll see when you look on the microscope. This is an image of prophase. You can see that the DNA is in these sort of thick strands. So it's been packed into these dense chromatids, and we cannot see a nuclear envelope because that's being dissolved. And you can see the spindle fibers that are attached to centrioles, which we also can't see in this particular picture. Then we move into metaphase. In metaphase, those spindle fibers are moving the sister chromatids around so that they're all in a line. And then in anaphase, the spindle fibers begin to shorten and pull the sister chromatids apart. In telophase, we have two distinct nuclei. You can start to see the cells splitting here, and it completes that in cytokinesis, which I don't have an image for of this particular cell. Here's a drawing that I think is lovely for showing the smaller steps between the stages. So we've got interphase here where everything's loose chromatin. This is just the nucleus. Late interphase, early prophase, things are starting to get packed up, getting packed up even more. Definitely in prophase. Now I'm starting to see a little separation here. Now we're moving into metaphase. You're trying to get them all in a line. Now we're in metaphase. Here's early anaphase where we're just starting to pull them apart. Later anaphase. Telophase where we've got two distinct nuclei. And then this last one is really just the beginning of anaphase, but showing you how the spindle fibers are arranged. Now, if we get a slide of where there's rapid growth, like a root tip or a blastocyst, which is like it's an early structure after sperm and egg meet. So we're doing lots of cell division to grow this new organism. Then we will see cells in every phase of mitosis. So here we can see interphase, a nucleus in interphase. Here's one that's in metaphase, sort of moving into anaphase. Here's one that's in later anaphase. It doesn't have all of them in this particular picture, but when you look on your microscope slides and move around, you'll see every phase for yourself.